form of carbon in the soil, and there is. There are two forms. One of them is the occluded, that's a UL, occluded light fraction. This is the labile fraction, occluded light fraction, and heavy fraction. This lasts for weeks or months. This lasts for decades. This lasts for thousands of years. So my question to the scientists is, my production of grass, removing carbon, producing carbohydrates, and also increasing durable soil carbon. In 2008, they did not know. So we designed a set of experiments to test this idea, if perhaps we could move carbon from the atmosphere and put it in the soil and see what happens. So there's a lot of difference between the types of carbon. Labile carbon is in the plant tissue. Occluded light fraction is when carbon in the soil starts to accumulate. This is good, this is humus. It's sticky, and it changes the water holding capacity. It changes the electrical properties. When there are increases of light fraction carbon, the electrical properties change, and now rainwater that was subject to gravity passing through the system, when there's increases of carbon here, rainwater becomes particles detained in a plant available form. So increases of occluded light fraction carbon automatically increase water holding capacity. And that's a good thing. And also heavy fraction carbon, this occurs, and it could be on the surface or a meter deep, anywhere in between. It's an opportunity, it's a matter of circumstance. So when a microbe eats plant tissue, for a moment, the free carbon that's released can actually be caught inside soil particles, and it's glued, and it's permanent. So heavy fraction carbon, occluded light fraction carbon, and labile carbon are the three times, different time cycling. So our question scientifically, can I increase these? 2008, nobody knew. How do we test this? The idea was to put compost. So compost, we put 1/2 inch of compost in December of 2008. So very thin, and we measured the results. And what we saw, within 6 months, we saw a response, and it's important to know, we did this scientifically, so we have plots. These are 25 meters by 65 meter plots, and then we have a control plot. So control and treatment. So this is science, and we replicated them 3 times. And then we took the whole experiment and we went up to the California Sierras. So we bracketed the entire California rangeland system and did the same experiment in both extremes. So we put 1/2 inch of compost on plots that had soil and moisture sensors to 30 centimeters. And Dr. Silver did a complete biogeochemical analysis to a meter deep in 9 locations. UC Davis, Dr. Val Evener, looked at all of the vegetation, so she knew everything about the plants. Dr. Silver knew everything about the soil system, and Dr. Creek, the other scientist, knew everything about the compost. This compost that we used is a biologically stable carbon-nitrogen complex. It's stable. This is the result of vermiphilic composting. So it has carbon, nitrogen, and beneficial organisms. We put 1/2 inch on top of the treatment plots in December of 2008. And by May of the following year, we went back and looked. Immediately for the vegetation, we saw 50% more grass, and it was higher nutrition. So it's better grass compared to the control. Because of the soil moisture probes in the water, we measured 26,000 liters more water per hectare compared to the control. So that water holding capacity was measured. It was significant and important. And for our purposes, measuring the carbon, we knew We removed all of the carbon from the compost. With the instrumentation, we measured the emissions, the CO2, nitrous oxide, methane emissions, and we subtracted all of the labile fraction. So we picked out all of the roots and burned off all the labile. Looking at just the two more recalcitrant fractions, we measured 2,000 pounds more per hectare. This is huge compared to the control. It worked. So we intentionally added carbon that ended up in a durable form within 6 months by adding compost. This was very exciting results. And so that was exciting. It was a big success scientifically. We've got several peer-reviewed published papers. We've created a protocol for this, which has been approved for carbon credit trading now. So ranchers like myself can do this additional work, add carbon from the atmosphere into the soil that's measurable, and then trade those carbon credits. So it's a good thing, very successful.
Where does the idea came from?
That's a great question, and we've expanded our research. But something amazing happened that we didn't see coming. That was 2008, and we didn't do anything else. We just let the grass grow. The next year, we went back and measured again. Another 2,000 pounds came in on its own with no further compost. And then the next year, and the next year, and the next year, it keeps going. So this is the most important story. Every year for 5 years, another ton came in and in and in and in. So it's 10,000 pounds of carbon in 5 years from 1/2 inch, one time. This is exciting, but how did that work? What's happening? Here's the question. So, as we know, photosynthesis is the photochemical process where carbon is transformed between carbon pools. So atmospheric carbon dioxide becomes photosynthetically derived biosphere carbohydrates, becomes rhizosphere durable soil carbon. It's good, it works. The pathway is the plant tissue. The plant is carbohydrates. The roots are carbohydrates. So now we have carbon in the soil as plant tissue carbohydrates. During different events, the plant sloughs or lets go of plant tissue. And it also, around every pore of every root, it exudes sugar. Tremendous amount of sugar is going into the soil to feed the bacteria, the fungus, all of the systems. It's feeding these living forms who then eat the plant tissue. Most of it respires, however, some of it goes towards permanent. And also the association with fungus, mycorrhizal fungi. The plant gives sugar to the fungus in exchange for nutrients. So nutrients come back, sugar goes out. So huge amount of carbon, huge amount of carbon, huge amount of carbon. And then the fourth pathway is the natural accumulation of plant litter, leaves, stems, manures, dead bodies, and healthy soil have organisms who pull this down. That's the pathway we primed by applying compost. That first application resulted in increased water holding capacity, which promoted more plant growth, which removed more carbon. And now the system is flowing. Very exciting results. This data set, when we take it and use a computer model, it says this will continue for 30 to 100 years from a single application of compost. Very exciting. And so far, the measurement is validating the model. So we have peer-reviewed science, we have a protocol, we have the modeling to show that this will continue, all the while producing more and more and more carbohydrates. Carbohydrates are sugars, starches, and cellulose. And when you add nitrogen, you get proteins. So the question is, how can agriculture be managed in a way to produce an abundance of carbohydrates and also increase soil carbon, and we've shown this method. So there are a bunch of questions. The production of carbohydrates, sugar, starches, cellulose, which create food, fuel, fiber, and flora. My clothing, this is organic local cotton. This is indigo, the plant. And so on my ranch, we now have the indigo production. And so we're making indigo. These pants were made from those plants.
Wow.
And the cotton. This is a hemp shirt. So there are ways now to provide fabric that results in increased carbon. We can make food that also results in more and more and more carbon being removed in the process.
Same system can use.
Yeah, exactly. And fuel, we're now experimenting with ethanol and methanol and biodiesel, all based on the abundant vegetation to produce liquid fuel, liquid energy, durable fiber, consumable food, and flora, which is medicine, pigments, lumber, things like that. So the proposal that we're now initiating is to satisfy the entire planet's material cultural needs in a way that removes more and more and more carbon. So with 8 billion people all making informed consumer decisions, we can reverse global warming. So it's working. We've done it. It's very exciting. The question is there enough compost? Right? And so there isn't now, but there could be. Anything that was ever alive can and should be composted. That's our position. Now, to develop this further, we needed to know of the available feedstocks. Can you make safe compost? Now in California, we have a state agency that regulates compost production to make sure it's safe. And to do that, we rely on thermophilic bacteria, which is naturally occurring. There's thermophilic bacteria on every surface. It's in the air, it's everywhere. And as a compost manager, I manage air, moisture, carbon, and nitrogen, and create the ideal environment for thermophilic bacteria to populate. They do, and everything does. But soon, as they respire CO2, it gets warmer and warmer and warmer. And in that heat, they destroy pathogens and weed seeds and things like this. So it's very important to do a thermophilic-based composting and get temperature when you're not certain that your feedstocks are safe. Now there are lower temperature composting, which is good. And if it's food scraps and things like this, that's a very appropriate. And there's also vermiculture, which is worms eating. This is good too for most safe feedstocks. But for us, with 7 billion and 8 billion people, there are also available feedstocks. Human waste is a very important under-recognized feedstock. So our next research project, working with Lawrence Berkeley National Laboratory, Dr. Gary Anderson invented a microarray, and it's this big, very tiny. It's no bigger than your thumbnail. But on this microarray at Lawrence Berkeley Lab, there are 1.1 million programmable sites for the DNA of all bacteria on Earth. So working with Dr. Gary Anderson and the state of California, and in my county, the Marin County Health Department, for 4 years now, we've been composting human waste. And using this technology, we've been able to show that we can destroy all pathogens with absolute certainty. And so we've done this, it works. Now we're currently composting pharmaceuticals. So we're looking at ibuprofen and Cipro, which is an antibiotic. And we have a specially tuned mass spectrometer at Lawrence Berkeley Lab that can see the signal of these pharmaceutical compounds in the compost. And we're watching the degradation of that compost through the thermophilic process. It's very exciting. Our next work will be looking at hormones and endocrine disruptors. So these are the products from birth control pills and other medicines and pharmaceuticals. When those go in the natural environment, it's devastating. And so it's our inquiry scientifically now, can we take all human waste, compost it, and deactivate chemicals, pharmaceuticals, pathogens, and create another feedstock to put out on these food-producing or fiber-producing soil systems. That expands the feedstock capacity, but it has to be done with certainty. So we're doing it.
Yeah, I mean,
It's very exciting.
it's very exciting. I just can't imagine that it's gonna